Okay guys, we're going to pick up right back where we left off. And so let's talk about the phloem and how the sugar moves through. So we said that right here, this loading part right here, this is active transport, right? Okay, so remember that requires ATP. And so if you think about it, we're stockpiling, we're moving against the concentration gradient. And plus, sugar is pretty big, so it doesn't just diffuse across the cell membrane. It actually needs to go through a transport protein, and then once it builds up here, it's like we still have to keep loading it in. So we have to keep adding ATP to get the sugar to get in here. But once it is in here, and it's in our sieve tube member, what's going to happen is just through pure osmosis, water is going to passively diffuse in. So remember, there's aquaporins, little tiny pores in all cell membranes that allow water in. And so water is going to follow the solutes because sugar is a solute. And so water is going to follow it, be attracted to the sugar, and what's going to happen is there's going to be a pressure buildup right here because we're just jamming stuff in here. So like a balloon, this is going to fill up and get pressurized. So let's mark that high pressure. Okay, so since there's a high pressure buildup here, anything that starts falling is going to have some force behind it. So the pressure that builds up here is going to squeeze the sugar down, kind of like a toothpaste tube, right? And so down here, though, we're going to unload the pressure. We're going to actively transport into the root cell. Okay, so the roots already have sugar stored in there, and they already have lots of starch and everything. So this is also active transport here. Okay, and once there's less sugar here, the water is not going to be as attracted to this area, so water is going to passively, by osmosis, actually go back into the xylem vessels. Okay, so let's make sure we mark this as xylem. Okay, and so the water is going to go back, and remember, water is going to creep up due to just pure passive attractions like cohesion and adhesion, right? And so water is going to go up, be attracted to the sugar being loaded in over here. The pressure is going to push it down. That pressure is going to be unloaded when we actively transport the sugar into the root cell. And remember, it's in the root cell that all the sugar is going to get turned into starch. When you put sugars together in a glycosidic linkage and keep doing it, and you can keep branching it out in big long chain, that's going to make starch, and starch is just going to be here. And then if the plant needs the starch later, if it needs some food, it's going to do the opposite. It'll actively transport that sugar anywhere the plant needs food. Okay, so that is how sugar moves through. We're going to move on to water, all right? And so for that, we need to understand the root cross-section. So you can see if we cut a little bit of the root and laid it flat right here, this is what we would see. And so this part right here is called the vascular cylinder. So that includes all the xylem and the phloem, right? And this layer right here, the pericycle. So your diagram looks slightly different. So I'm going to need you guys to kind of look at it and make sense out of it. And it's not a bad idea to color in these cells. So the pericycle, this is what gives rise to the lateral roots, okay? So lateral, meaning sideways, roots grow from this layer. Okay, now the next layer right here where you see this kind of red band going through, that's called the Casparian strip. The Casparian strip is this red part, and that red part goes through this layer. And so that layer is called the endodermis, and the endodermis protects, the whole job is to protect the vascular cylinder. Okay, so the reason why we have this layer here is because if there are any viruses, and there are viruses that protect or that target plants, if there's any harmful bacteria, if there's a fungal infection, the endodermis is going to protect from that fungal infection or whatever that infective agent it is. It keeps it from getting into the vascular tissue, because if it gets into the xylem or the phloem, that's going to travel straight through to the leaves. It's going to travel all over the plant. So think of the xylem and the phloem as kind of like the veins and the arteries of the plant, and so we don't want anything bad to get in there because then it'll spread. Okay, so it's to help keep everything localized that could be harmful. All right? So here's what I mean by that. There are two routes where water can get in. Symplastic means goes through living tissue, okay? Okay, so what that means is the water actually enters into the cell, okay? And you can see it's going through the middle of the cells. So it's going to be transported. And remember, plant cells have these things called plasmodesmata. They're little tubeways that connect all the cells so water can get through. So if it's going through this plasmodesmata, through the middle of the cell, it's just going to keep going through and it's going to enter. Now, to get it in here, it had to cross the cell membrane. The cell membrane is semi-permeable. So nothing bad can get through. Well, sometimes it can, but most bad things cannot get through the cell membrane. So a lot of that bad stuff gets filtered out. Now, the thing is, though, water also travels just along the sides of cells in the cell wall. That's the apoplast. So it never actually enters living tissue. So water is going through cell walls only. Okay, so remember, the cell wall is just a layer of carbohydrates on top of the cell. And so if it's just attracted to that part, it's going to kind of travel along the sides of the cell. It never actually enters the cell. It never passes into a membrane, okay? And so the problem is when it gets right here, it can't just stay that way. It actually has to enter a cell. So you see right here how it's going through the middle? It's actually passing into the symplastic pathway there. Okay, now this layer right here, the Casparian strip, is waxy. So water is just going to hit a block. If it tried to go this way, it would get rejected and come back out. So this Casparian strip is going to force water into the symplast, and therefore we can filter out anything bad that might be in the water. Okay, so that's that. Now let's see, what is going to cause water to actually enter here? All right, and water movement, that's osmosis, and that depends on, well, our old friend water potential. Okay, so remember, water goes from high water potential to low water potential. Okay, so the root's water potential must be lower than the soil's. Okay, so if water is going to move in, this water potential must be low. And so remember what lowers water potential. So remember, solutes lower water potential, okay? And solutes make this area more negative, okay? Make it lower. So water is going to want to enter and be where the solutes are. And so that's kind of what happens here. So part of the reason water enters a plant is just through just pure osmosis. There's just going to be solutes in the root, okay? So you can see in the root, well, they're calling it the lower trunk here, but basically it's the roots, negative 0.6, whereas the soil is negative 0.3. So from high water potential to low here. So water is going to enter. And you see, as you go up the tree, the water potential, there's a gradient there. So water is going to keep wanting to go from high to low water potential. The outside air has the lowest water potential, so water's eventually just going to evaporate. Okay, so as you can see here, water is evaporating out of the stomata and into the air. Okay, and you can see here, cohesion and adhesion is driving a lot of this. All right, and anytime one of these water molecules leaves, it's going to pull this whole group of water molecules up. So you see, it's kind of like a column. And anytime one evaporates, it kind of leaves this vacuum here. And so more will come and take its place. And that's how water just kind of continuously moves through. All right. Let's go back. Oops. Okay. So let's look at these things. So osmosis, like we said, water is going to enter the roots through osmosis because there are solutes there. Okay. So there's solutes in the root. Water is going to go in to follow those solutes. once it's in, there's going to be quite a bit of tension there because the water molecules are not only attracted to themselves, they're attracted to the outside wall of the xylem. All right, and like we said, anytime water evaporates, it's going to pull the whole column up with it. Guttation is a phenomenon where basically water is coming up and not evaporating off the surface, and so it'll form these water droplets. So it's different from dew that comes from the atmosphere. So water in the atmosphere kind of condenses on the leaf. This is actually coming from the root. So root pressure is pushing this up and creating these really pretty droplets like that. All right, so all these kind of contribute to water movement in a plant, okay? And in our next lab, hold on a second. Oops. Wrong way. Excuse me, guys. There we go. All right, so in our next lab, we're going to be looking at this thing. It's called a potometer. Okay, and so we're going to set this up. This is a pipette. We're going to fill it with water. We're going to fill this whole thing with water and stick a plant in there. And as water evaporates, our water level is going to go down, and we'll be able to measure excuse me, exactly how much water is evaporating out of this plant. So we can put it under different conditions and see what happens. Okay, so here you can see the water level has gone down due to transpiration. So transpiration is the technical term for water evaporating out of a leaf. Okay, and so the more water evaporates, we're going to see our data show an increase in that. Okay, so we're going to measure it over time, and we're going to measure transpiration here. Okay, and so we'll be able to see how quickly it goes. All right, and the more transpiration there is per unit of time, the steeper this slope will be, and we can calculate the slope and calculate the rate and all that. All right, so here's a little cartoon diagram of transpiration. You see the water, every time it evaporates, it pulls up the water behind it, and you guys are going to watch another video that goes over transpiration in a little bit more depth, and you're going to try to set up your own potometer. Okay, so good luck with that, and I'll see you guys next week.